Hi, everybody. So last time we talked about the purposes of mitosis, of asexual cell division. And mitosis makes identical cells from the parent cell. They have genetically the same DNA. So that means that mitosis has to be very careful to copy all of the DNA and then make sure each daughter cell gets an exact copy. We don't accidentally want two copies of the same genetic information going to one offspring and the other one not getting that information at all, because then one cell may have the information about how to make the organism completely, and the other one won't have all the information it needs to function. So every time a cell divides, all the information has to be copied and then divvied up. Now, that information is DNA, and it's found inside the nucleus. And so during mitosis, we are going to have to pay really close attention to the nucleus, the genetic material inside of it. All that DNA, all that genetic information has to be copied and then has to be very carefully separated so that one copy goes up and one copy goes down, and that we don't have two of the same copies going in the same direction. It's a very, very careful process. And to do that, to talk about that, I want to make sure that we've reviewed all the structures that we're going to be talking about and that you're familiar with them. So first of all, when we talk about things moving around the cell, we use the terms poles and equator as if it was the Earth. So if something is moving toward the poles, that means it's moving toward the either end of the cell. If it's lined up across the equator, that means it's across the middle. Now, I may use the term middle or equator. I might use ends or poles, but you'll see those terms from time to time, and I want to make sure that you know that we're talking about just like a globe. So at either end is the poles, across the middle is the equator. Now, there are some key things we have to pay attention to. Because mitosis is copying the DNA and then divvying it up, we want to watch the DNA, so we have to know where that is. And there are some things, once we copy this, some of it's going to go that direction, some of it's going to go that direction, and we're going to have some things in the way. The nuclear membrane around the outside is in the way. The nucleolus is in the way. And so those are going to have to be removed. Like, we have things to keep track of. So we have, first of all, the nucleus, okay? And inside the nucleus is the nucleolus. And also inside is the DNA. Okay, so those are things we want to keep track of: the nucleus, the nucleolus, and the DNA. These little star-shaped organelles are centrioles. Now, you may remember that centrioles are found only in animal cells, but they're important for cell division, for cell reproduction. And that's what we're talking about now. And so that's going to be important for us to keep track of. So we have the centrioles in animal cells only. All right. And finally, there's this nice picture that kind of gives us an overview. We've got the nuclear membrane, we have the DNA, we have the nucleolus, and then we have the centrioles. Sometimes you'll see it referred to as centrosome. That includes some other parts. For us, it doesn't matter. You can use the word centriole just fine. And if you see centrosome, read it as centriole. Okay, so those are the structures that we're going to keep track of. Now, there's a couple other terms that I want to make sure that we have before I can talk about how mitosis happens. And that is the forms of DNA. Now, ordinarily, when you look at a cell in the microscope, the entire nucleus is just going to sort of be shaded all one color. It's just going to be solid looking. And that's because the DNA, even though it's a string, is so fine and so jumbled up and all over the place that it just looks solid. That's how DNA is through most of its life, most of the cell's life. And it's the perfect form for it to be in to copy. It needs to be in a jumbly mess. Think of a litter of kittens that have gotten a hold of a whole bunch of yarn, and the yarn is everywhere. It's just sort of scattered. The cell likes it just like that for copying. So anytime the DNA has to be copied, it's going to be in this jumbly mess form, and that is called chromatin with an N at the end. Okay, chromatin. And that's the jumbly mess. A litter of kittens have gotten a hold of it. It's all over the place, perfect for the cell to copy. On the other hand, if you think about this, once that's copied, we have to take a copy and send it in one direction, and then take its identical copy and bring it in the other direction. We have to separate these identical copies, and we have to make sure that we don't accidentally put two of the same copies in one direction. We have to make sure that if a copy goes this way, a copy goes that way for every single separate chromosome in the nucleus. Now, when it's chromatin, think jumbly mess, kittens have gotten a hold of all this yarn. That is a lousy way to separate and make sure that you have identical copies going opposite directions. So chromatin may be great for actually copying it, but it stinks for separating it in an orderly way and making sure that each daughter cell gets a copy. And so before it divides, it will switch over into organized sticks. It'll take this big jumbly mess and wind it up into sticks so that they can easily be separated. And those sticks are called chromosomes. So you can have a chromosome that's made up of one and another copy. They're identical to each other. And now that they're sticks, when the cell goes to separate it, it can be sure that the copies get separated. And one cell, daughter cell, gets one, and the other daughter cell gets the other. And so chromosomes are good for dividing it up. Chromatin is good for copying. Chromosomes are made up of, can be a single stick, or it can be with its copy. And when it's with its copy, it's called a chromatid. Very confusing. We have chromatin, jumbly mess, kittens got a hold of it, good for copying. Chromosomes, those are sticks, easily moved around, but not good for copying. And anytime that you have an identical copy with its mate, it's called a chromatid. To see what it looks like in the cells, that is chromatin. That's a cell that's busy copying its DNA. And at this point, it's all copied. It's now in chromosomes so that it can be separated into two daughter cells, chromosomes and chromatids. And that is the introduction that we need before we can talk about how cells divide and make identical offspring. Key takeaways: jumbly mess is DNA in chromatin. It's excellent for copying the DNA, absolutely essential, because during mitosis, we are going to separate identical copies and make sure that each daughter cell has the full set of genetic information from the parents.